Alrighty, folks. I'm gonna try to get this going. Is everything all right? Is the slideshow showing up all right, or was it just showing right then? Actually, before we get going, little heads up here. Uh, this is technically Unit One, biology. Uh, it is very boring. It is very vocab dense. I've got like some charts in there. Got a couple of diagrams of like the brain and stuff. So a lot of that stuff is really hard to understand. But hopefully, all of you uh, are in psychology right now. You've kind of already done this, so it's more of a review. But I just know shouldn't go over 2 hours. But I will let you know if we will because it very well could happen. Assuming everything will work right, I'll go. I'll go and get going. AP Psychology. This is the Unit One review. Technically speaking, some of you may have had it where biology was technically Unit Two. I had it that way this year, but just for reference, we will cover the first five major units, as in biology, cognition, learning and development, social psychology, and then mental and physical health. And then I will go back at some point, probably that Saturday or Friday before the exam, and give a brief overview of the FRQs and what is Unit Zero. It was Unit One for me, just research principles, that kind of stuff. So it's all very important. Also ethics, all that stuff. But if everything's all right here, go and get rolling. And before I say anything actually psychology, just know that I'm a volunteer. I don't get paid for this. I made this on my own time. I'm volunteering my time. This does not guarantee any grade. You just because you attend here doesn't mean you're gonna get a 4 or 5. This is simply a review material. This is supposed to help you. But here we go. So our first subsection here of biology, technically Unit 1.1, interaction of heredity and the environment. So this essentially boils down to the whole nature versus nurture debate, whether heredity plays a more important role in your behavior mental processes, or if environmental factors do. Heredity or nature, internal factors, you're predisposed to it, genetics, that kind of stuff. Now, the environment or nurture is external factors, experiences such as family influence or upbringing. It's just outside forces that are not internally predisposed. Yeah, and the reason the slides look like a watermelon, as I was told, is because that is what the course exam description title page is colored. It's kind of weird, but it works. And of course, these slides are listed on the calendar and also in the chat. If you want to go back and look at them, I don't care. And then, technically, also included with heredity and the environment is the evolutionary perspective of psychology. Essentially, just boils down to natural selection. It's essentially behavior and mental processes survived and were passed on based on survival and reproduction. Basically, the most beneficial behaviors and mental processes, or more those individuals with them, are more likely to survive, reproduce, pass them on to the next generation, so far. And then how we research the effects of genes on behavior, the FAT acronym, through family studies, adoptive studies, and twin studies. All right, I didn't put a mini quiz there. Um, and then since 1.1 is not really anything special, it's a ready environment. Let me break right into 1.2, starting with the nervous system. So the nervous system has two main branches, those being the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which is everything else involved in the nervous system. Um, Basically, all the nerves throughout your body, excluding the spinal cord of the brain. And then there are a number of sub nervous systems. Um, so you start here with the autonomic nervous system, which is controls involuntary actions, its bodily functions, breathing, your heart beating, stuff like that. Then the uh, and then falling under the autonomic nervous system is the sympathetic nervous system, which is it basically activates fight or flight. It mobilizes the body, increases heart rate, breathing, all that stuff. Stops digestion. And then also under autonomic is the parasympathetic nervous system, which is which is the counter to the sympathetic nervous system. It calms the body down. It slows you down. So basically, after the fight or the flight, your body has to calm down, slows heart rate, breathing, that kind of stuff. And then the somatic nervous system, which is which both autonomic and somatic both technically fall into the central nervous system. And the somatic nervous system is voluntary actions, walking, muscle movements, that kind of stuff. It's pretty no. You just need to know that we study that stuff through family studies, adoptive studies, and twin studies. You just need to know how we study it. You don't need to know like specifics on it. Just ignore this. So just think to yourself. Don't put anything in the chat. I'm not going to make you do that. So just think to yourself. You should know this. What is heredity, and what is the difference between the autonomic and somatic nervous systems? Just so we a couple of minutes. And just I'm not making you put anything in chat. Just like if you want to, you can. I don't care. And studying there, yes, but autonomic really is it movements technically. It's like bodily functions essentially. So your heart beating, you breathing, just stuff you do automatically, and it's automatically controlled. That's in effect what it is, simply. So rolling along here, starting here with 1.3 and all the way through 1.5 is kind of a lot, like a lot. Um, so specifically here, 1.3, the standard neurons, how they fire, their connections, neurotransmitters, hormones, psychoactive drugs. It's got a bunch of stuff under it. but we're starting here with the neurons. So they're neural cells located in the brain, spinal cord, nerves. They perceive and send information. There are several types, and there are also more, but these are the ones you need to know. Sensory neurons, which bring in stimuli from the senses. Motor neurons, which send information to muscles. Interneurons, which are in the brain, and they connect sensory and motor neurons. And then afferent versus efferent. Afferent carry impulses to the CNS, and efferent carry impulses away from the CNS. And CNS is a central nervous system. Going along here, and there is a diagram on the next slide. Um, so the structures of a neuron. Now this is a lot right here. So essentially, so communication starts with the dendrites, which receive neural information. They bring it in, they send it into the soma, which contains the nucleus. I'm pretty sure we all know what a nucleus is. It's genetic material. It's the brain of the cell. The soma, which is basically just the cell body, directs any movement or reactions of the cell. Then you have the axon hillock, which connects the soma to the axon. You have the axon, which is kind of like a wire. So it's like a wire carrying your charge, carries neural information. Then you have the axon terminal buttons, which then send the information slash message to the next neurons. And then the myelin sheath is what insulates the axon. So think about it like covering over a copper wire, protection essentially. It also serves to speed up impulses, and then glial cells. It's spelled wrong. Which nourish, protect the neuron. They're kind of just floating around. They're not necessarily a part of the neuron, but they are important to a neuron's survival. So essentially, what happens? I'm putting a diagram here. So essentially, so a message is received over here in the dendrite. It is then passed through the soma, through the axon hillock, down the axon, the terminal buttons, and then out to the next neuron. And just think of this neuron here being in a chain of like millions, or probably even billions. I think a lot. This is a tiny little thing and a massive operation. It's glial cells. It's just GILAL. I put it on the wrong end. That's in the chat as well. So keep it rolling here. So when you come here to a neuron firing. So you're gonna start here, stage one, step one, which is resting potential. It is a neuron's normal state. Positive ions are on the outside, negative ions are on the inside. Then you move here into threshold, which is the minimum amount of stimulation you need to trigger a charge. So basically, so a neuron starts rest potential, then when it reaches threshold, then it will fire, which is an action potential, which is that's the name for the electrical charge that travels down the axon, sends the message, and then four is technically depolarization. Positive ions enter and open the axon, which then sends out the message through the axon. And then you have the all or nothing response, which is so the neuron fires with the same intensity or not at all. There is no partial fire; it either fires or it doesn't. Then you have the refractory period, which is the time when a neuron cannot fire and returns to resting potential. So let's see if I can find real quick. Yes, there is. So it was brought up in my psychology class. It's here on the Google page. Is so you can so essentially a good analogy for a neuron firing is a toilet flushing. I can't give you the whole thing here, but essentially what it is for resting potential. The toilet's just sitting there, normal, not used. Threshold, something goes into it, some bodily discharge, and then the exponential is that stuff going down. Depolarization is when I believe if I'm correct. I don't know. Depolarization essentially when you flush it. The all or nothing response means you either flush the toilet or you don't. You can't partially flush the toilet. And then the refractory period is when the tank has to fill up again, so you can't flush it again. If you want to, you can Google that. I'm sure there's a video on it somewhere. It's just a neat way to summarize it. And then so neuron fires, and then but then what does it do? How does it communicate? So so a neuron communicates through what is called the synapse, which is the gap between each neuron and the space of which a neuron communicates over. The vesicles are sacs within the terminal buttons that hold and release neurotransmitters. Reuptake, which it's basically sending, so the neuron that sent the neurotransmitters reabsorbs whatever ones are left over in the synapse. And the neurotransmitters are chemical messengers that send info between neurons. So think here, I'll jump back here to this. So essentially, so the neurotransmitters are coming out of these terminal buttons, and then they are going to the next cell's dendrites. So think there's another neuron down here on the bottom right. The dendrites are right there. So neurotransmitters will leave the vesicle from within the terminal button, and then travel to the next neuron's dendrites, and it starts the process over again. And then here we are. So, which neurotransmitters do you guys know? I'll tell you if they're required or not. There are a lot, but not all of them are required, of course. Just put them in the chat if you want to. Oh, never mind. Yeah, apparently that was spelled wrong. Dopamine, serotonin, GABA, norepinephrine. Yeah, those were actually all required neurotransmitters. Pretty good. Because I know for a fact most of, if not all of you, have already taken most of psych. One moment here. And then whoever that was. And whatever, because it blocks your name because I'm recording. Um, the way it had on the slides is how you spell it. I just mixed it up. So for neurotransmitters, we have crappy little charts. So so the required neurotransmitters. So there's four on this side. I think there's five on the next slide. So you started with acetylcholine or acetylcholine, however you want to say it. So what it affects is learning, memory, and muscle action. Too much of it can lead to depression, and too little can lead to dementia, and Alzheimer's. Dopamine, pleasure, mood, emotion, and attention. Too much is schizophrenia. Your brain becomes too active, so you start seeing things. Too little, depression, tremors. You're depressed. You don't feel pleasure. Boom. Endorphins, which is pain relief, and euphoria. That does not affect too much, too little. Yeah. GABA, which is the main inhibitory neurotransmitter. Too much, you feel relaxed or sedated. Too little, you're anxious. And then the other four here: glutamate, which is the main excitatory. Uh, Too much can lead to migraines and seizures. Too little, psychosis or coma. Norepinephrine, also known as adrenaline. Panels, alertness, and arousal is also excitatory. Too much is schizophrenia. Too much adrenaline, you're too active. Too little, depression. Um, Serotonin, which is primarily mood regulation. Too much is heart issues, confusion. Too little, depression and anxiety. And then substance P, which is pain transmitter. It doesn't affect anything else other than pain. It basically what tells your body to feel pain. Keep on rolling here. Let me come here into hormones, which is they're essentially neurotransmitters, but they're a little more specific. So the correct five, yeah. So the five hormones you need to know: adrenaline, which is basically does the same thing as norepinephrine, it's just not a neurotransmitter. So think of it: hormones are neurotransmitters for anywhere but your brain, because neurotransmitters are only passed by neurons. Hormones anywhere in your body. So adrenaline is primarily responsible for fight versus flight. It mobilizes your body. It excites it. Increased breathing, heart rate, that kind of stuff. Melatonin, which is essentially your body's natural sleep hormone. Um, if you and then if you know the medication melatonin that helps you sleep, and it induces sleepiness. Then you have ghrelin, which is the hunger hormone. It stimulates appetite and it promotes fat storage. And then leptin is also a hunger hormone, but it inhibits appetite. It indicates that enough fat has been stored. It's actually released by the fat cells in your stomach. And then you have oxytocin, which is commonly known as the love hormone. It enables bonding, empathy, and trust. Those are the five you need to know hormone wise. You don't need to know what was it? I forgot. There's some other ones that are mentioned. But you don't need to know them for the exam. Yeah, I think the biggest difference between neurotransmitters and hormones, they will do similar things, but neurotransmitters are only passed and only understood by neurons in your brain. hormones can basically be interpreted by any body part. I think. Fact check me if I'm wrong, but neurotransmitters are neurological transmitters. They only work with neurons. Hormones can go anywhere. And what HWA is saying in the chat is good, valuable information. It's yeah, hormones, of course, obviously take longer because neurons are they're tiny, they're tightly packed in there, and they fire like never seen before. They send messages very, very, very quickly. And for example, hormones are typically released by the pituitary gland, which we'll cover later. But essentially, and also think growth hormones. You grow slowly; those hormones promote growth. HWA. Are you talking about drugs, or because the there's nothing in the course prescription that mentions like a natural uh, inhibits your blood. Yeah, those are a little later, I think, and they're in this like subunit. But yeah, I have like a slide for each of them, so don't worry. And then I don't care. Yeah. So when you guys think of a psychoactive drug, what do you think of? Can you put some ideas in the chat? Yeah. Cocaine. Yeah. LSD is a drug. Yeah. It's psychoactive. Yeah. Basically, a psychoactive drug is anything that affects your brain. It affects your psychology, essentially. Alcohol is a psychoactive drug. Stimulants, yes. Are you on the slides or free? Caffeine, very fun. And I think just about all those suggestions were like perfectly right. Or at least all count. So rolling right in here. So psychoactive drugs are so there's two main types: agonists, which mimic neurotransmitters and they encourage neural firing, and then antagonists, which block neurotransmitters and they discourage neural firing. Now, those subtypes, you just need to know what they are. You don't need to apply them to any specific drug. And I have like four different categories here for drugs. Rolling right in here, we have stimulants. They're going to increase neural activities, such as caffeine and cocaine. Two very good C's. No, don't do cocaine, kids. Um, so some physiological effects are increased heart rate or increased energy, increased heart rate, headaches, irritability. They're going to ramp your body up. They stimulate you, they energize you. And then psychological effects, poor decision making, and suicidal thoughts. Yeah, basically, if you have too much, it can drive you insane. And then depressants. These decrease neural activity. They slow you down. You don't feel anything. Alcohol and methamphetamines. So their physiological effects going to slow motor reflexes. You have drowsiness, slurred speech, and then psychological effects. They basically cause you to not feel, decreased memory and concentration. You don't remember anything, and also mood swings. This is why commonly alcohol is used to get rid of or drink away pain because it decreases your memory and concentration. You don't remember the bad thing that just happened. And then a little thing here is so. Um, yeah, so basically, so if anybody here has ADHD, I had to double check myself there. Anybody here has ADHD, you probably take Adderall, which helps you to concentrate. So, so essentially, so methamphetamine is a little different. So it's meth than what is the subcategory of amphetamine, but Adderall is a amphetamine. So technically, Adderall is meth, but not really. Just an interesting little fact there. And memorizing neurotransmitters, that's a little hard. I would say best thing you can do is I, I just say flashcard with the just flashcards with the transmitter and the events. That's about all I can give you. Neurotransmitters, there's not really a way to abbreviate or shorten it because there's eight of them or nine of them. Uh, the best advice for studying for psych, um, I'd honestly just say if you struggle on your normal tests, vocab, 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 vocab. Make sure you memorize your vocab because essentially what happens is if you don't know the vocab term, you won't know the answer to a multiple choice question. And also make sure you know how to answer FRQs, but that'll be addressed later. Uh, Short crab there. Um, if you don't have anything, I would say get to cranking out flashcards and just start doing them over and over again. You maybe do it make different sets for each unit. Do like a set a day, and then just try to get into your brain in the month you have. Mostly, well, essentially the multiple choice is basically just vocab. And then I'm assuming over the year you've been doing the FRQs, the EBQ and the AAQ. If you haven't been, I'll be doing a rough overview like before the exam, probably that weekend beforehand. But I would say look at practice ones, do them, and then figure out how you do them because there's a very specific formula to do them. That's yeah. Um, I would say for vocab, I'd say yeah, the FRQ just changed. It's the EBQ and the AAQ format. Yeah. 10 days for what do you mean for five? For 5 units? Oh, 10 days. Yeah, I know. I see at this point. Hope for a 3, maybe you can get a 4. If it. If you've been doing vocab, then I mean, because that's basically so basically what my teacher does. So what my teacher does for psych is his multiple choice questions are basically AP multiple choice questions. So you do it throughout the entire year. Then again, I'm sure guarantee you every other teacher is not like that because I've heard some horror stories about AP teachers. Oh yeah, especially well. So there's your four main categories of psychoactive drug. Or actually, there's two more. But real quick, before we go any further, if you guys want to go on your free time after this, I recommend you check out. It's the University of Utah or Utah. I don't know what it is, but it's essentially this little thing where like there's like different mice that are like addicted to a drug, and it, like tells you how the drug works and like the category. It's actually really cool to look at. If you have time, it's pretty interesting. It's essentially a mouse party, and they're all hooked on a different drug, and it's pretty weird, but it's interesting. It gives you a lot of information about the drugs. Okay, I'm gonna keep rolling. We still like 40 slides, guys. Um, so hallucinogens, which cause distortions and perceptions. Marijuana and LSD, they do the same thing, except LSD is synthetic, and marijuana is natural, quote unquote. So physiological effects going to be an increased heart rate, impaired coordination. You have intense feelings. You can have visions. Psychological effects: fear, depression, paranoia. You start seeing things because you're hallucinating again. Basically, just they're called hallucinogens because they make you hallucinate essentially. And then the final category is opiates, opioids, whatever you want to call it. They primarily act as pain relievers, such as heroin and morphine. The physiological effects, de- excuse me, decrease pain. That's what they're designed to do. Cause drowsiness and lethargy. They slow you down. They calm you down. Psychological effects. They are highly addictive. They cause euphoria, depression. Basically, if you aren't given one of these in medical sense, you're gonna be hooked on it. And then a little overview here of some psychoactive drug terms. So tolerance, which is the diminishing effect of a drug over time. Your body adapts, your body tolerates it, and your body needs more to get high off it. So essentially, over time, your body builds up tolerance, which means the next time you need to take more and more and more and more to get that same high. And then addiction, which is a compulsive pattern of drug use, comes with both physical dependence, which is a physiological need for a drug like your body needs it to survive, and the psychological, which is a mental and emotional need for a drug. Trying to cope with your issues, you need it to cope. And then withdrawal, which is discomfort after discontinuing drug use. I'm pretty sure we all know what withdrawal is: aches, fever, chills, depression, anxiety. It's basically your body saying, basically, let's say you're addicted to heroin, so your body's saying, "Where the hell is the heroin? I need it," and it causes those physical symptoms, which it obviously sucks. All right, psychoactive drugs. I don't care about that. So we jump right in here, 1.4, with the brain. So we start here in the brain stem. It's going to regulate autonomic functions, those automatic bodily functions, and it relays information between the spinal cord and the brain. Essentially, your brain's your control center, and your spinal cord's a little relay station. Your brain sends it down to the spinal cord, and your spinal cord has all the nerves, and they send it out to your entire body. The brainstem contains the medulla, which controls breathing and heart rate. The pons, which controls sleep, waking, and relaxing. And the very back of your brain, not part of the brainstem, is the cerebellum, which is balance and muscle coordination. It's basically, if you need to identify it, it's a mini brain on the back of the brain. That's what it basically is. And the midbrain, which only houses the reticular activating system, which runs along the rear of and part into your midbrain. It's going to transmit sensory messages, and it typically controls arousal, attention, and sensation. And it's responsible for the sleep-wake cycle. That's what wake. That's what he tells you to sleep. What tells you to wake. Yeah. I'll jump here real quick. Okay. And then the forebrain, which is the big part. So it basically contains the entire limbic system plus the thalamus, which is not part of the limbic system. So the thalamus, which is the only non-limbic part of the forebrain, it transmits sensory and motor information. It is perched on top of the brainstem, so it essentially takes what the brainstem gives it, sends it out to the rest of the brain. The brain sends what it wants back, and then it sends it back down to the brainstem. And then the four limbic system parts, to the amygdala, which controls emotions, primarily aggression and fear. It's fear emotions essentially. Then you have the hypothalamus, which controls hunger, thirst, body temperature, and sex drive. It's basic bodily functions. The hippocampus, which is primarily responsible for your memory process. When you think memory, think hippocampus. And the pituitary gland, which controls the endocrine system, which we do not need to know the endocrine system, and that is what controls hormones essentially. And here's your two diagrams. I'll keep rolling here. Here we come now. Don't do that. Um, so come here to the. So basically, the main part of the brain, which is your cerebral cortex, is split into two hemispheres. They each have different functions, but they work together. They are connected by the corpus callosum, which is a fiber network that connects two hemispheres and allows them to communicate. Then the cerebral cortex is the majority of your brain. It contains an outer layer of wrinkled ridges and valleys. It contains motor, sensory, visual, olfactory. It contains basically, you think about anything, you think about moving, your cerebral cortex probably contains something that involves it. And then come here to the lobes of the brain. So there is the occipital lobe, which is in the very rear above the cerebellum. It processes visual information. That's all it does. Occipital vision. Then the temporal lobe, which is on the sides of the brain there by your ears, and it processes auditory and linguistic, so speech information. Your parietal lobe, which is the upper back part of the brain, which is where the association areas are. It primarily controls your sense of touch. And then your frontal lobe, which is the majority of it, it's located in the front of the brain. It's the frontal lobe. It controls executive functioning, motor, linguistic processing, a bunch of stuff. And that's a chart with all the locations. Going right along here. So, within the brain, there are certain association areas. So, the two in the frontal lobe are the primary motor cortex, which controls and processes movement. It controls motor. You have Broca's area, which enables speech production. The parietal lobe contains the primary sensory cortex, which interprets tactile information, pain, pressure, and temperature. The primary auditory cortex and the temporal lobe, which processes hearing. Then you have the primary olfactory cortex, which is in the temporal lobe. It interprets what we smell. Olfactory is the sense of smell. Wernicke's area, which is in the temporal lobe. Speech comprehension. Wernicke's is comprehension. Broca's is production. And the primary visual cortex, obviously in the occipital lobe, and interprets what we see, size, color, light, and motion. So essentially, in the brain, there are four lobes, of which each contain different association areas of a certain sense or control. And we jump right in here um, to plasticity, which is the brain's ability to change with learning. It occurs after damage or a new experience. It builds new neural pathways, expands your brain. Not necessarily expands your brain, but it expands your brain. And then we have split brain theory. Essentially, this is basically when the two hemispheres cannot communicate with each other. The corpus callosum is cut. Two hemispheres, they work separately, and this is actually a treatment for somebody with really bad seizures. It's been found if the corpus callosum is cut, seizures are more minor in nature, especially someone in a major seizure issue, whatever. Um, Essentially, it cuts off the ability for the hemispheres to communicate, which thus makes those seizures less severe. So your left brain, which is more verbal, analytical, and orderly, and the left hemisphere controls your right leg and right arm. Your right brain, which is more creative, artistic, and imaginative, and that controls your left side. And I may as well show you this video clip. Um, let's see here. You know if you got. This is Stacey. All right. Actually, I won't show you this one. You want to go back and watch it at some point after you can. It's a case study of Phineas Gage who had a railroad spike through his brain. It essentially just tells you about how different lobes control different things and how a railroad spike through his head changed him because he survived. Actually, it's just a little interesting thing. Why not? Or are you back over? Okay, we're here. All right. So then we have how we study the brain, which so basically what used to happen was surgical procedures, surgical removal or destruction of a part of the brain. It examines behavior changes and infers functions. This was done before we even thought about what ethics were. Essentially, you would take a person who was considered to be mentally insane, obviously, pretty much most of the time, had a minor mental illness, something like that, and you basically cut out part of their brains and basically watched what that did. You didn't care about the person, and that was found to be very unethical and very inhumane. So now we have new ways of studying the brain, which there are a number of different, basically comes through technology, number of different machines slash scans that help us to analyze the brain. Another way they did a surgical discovery was through lobotomies. Another fun topic that I won't go into. So we have an EEG, which is essentially a bunch of sensory nodes placed on your head. They examine brain waves and determine brain activity. They are used in diagnosing epilepsy, brain tumors, sleep disorders. An EEG is typically used during a sleep study, which can help identify different sleep disorders. And then a CT scan, which is which is able to see structures of the brain, and it also determines damage, brain injury, tumor, strokes, that kind of stuff. There's two pictures there. Left side is the EEG, bunch of sensory nodes placed on your head. And the right side there's CT scan machine and then a scan, just for a little. And don't mind the voice cracks. And then we roll in here to the last three. So you start with a PET scan here, which is going to see brain activity during tasks. It, and also essentially, so a PET scan is one of the ones where they will inject um, glucose, some dopamine, something like that. And then, so while you do tasks, it's able to see what areas of the brain are using those resources to determine what it's doing, and help to diagnose tumors, infections, strokes, Alzheimer's, Parkinson's. We have an MRI, which is MRI, big magnet spin pictures of brain. It shows brain structures and surrounding tissues. Diagnosis can be used to diagnose tumors, cysts, infections, multiple sclerosis, dementia, stroke, that stuff. And then you have an fMRI, which is a functional MRI. It shows activity in basically MRI, but while you're doing cognitive tasks. And it is used to determine effects of strokes to see what parts of your brain are affected. Let's say there's a PET scan, right side there's an MRI. The bottom PET scan is kind of interesting. All right, now we jump here into 1.5, which is sleep. So we have the first topic here of consciousness. So when you are conscious, it's wakefulness and you are aware. Preconscious, which is memories and knowledge. Unconscious, which is your suppressed desires, feelings, ideas. And then sleep, which is your body and brain at various stages of rest. So you get in here, circadian rhythm. It's a biological clock that regulates our body, hunger, thirst, sleep, wake, that kind of stuff. And then you have certain brainwave patterns here. So beta waves, you are awake and alert. Alpha waves is like rest, you're relaxed, you're drowsy. Theta waves are present during late sleep, and then delta waves, which are present during deep sleep. And we come in here to the sleep cycle. So every sleep cycle begins with non-REM sleep one, which is when you start to fall asleep. Your theta waves appear, and you have hypnagogic sensations, which is basically just visions. You roll into non-REM two, which is light sleep, and sleep spindles appear, which are sudden bursts of activity. It can be seen in EEG. You have non-REM three, which is deep sleep, where delta waves appear, and then you have REM sleep, which is also known as paradoxical sleep. Dreams occur here. You see things, and there's a lot of activity within your brain. EEG shows that kind of stuff. And essentially, the sleep cycle goes non-REM one, two, three, REM, and then starts back over non-REM one, back down again, back up again, back down again. So basically, it goes non-REM one, two, three, REM, and then non-REM one, two, three, REM. And then so through the night, so basically as sleep time becomes longer, non-REM decreases while REM sleep increases. So over time on this chart here, you can see the later it gets, the more and more REM sleep there is, and that's a sleep you want. You don't want non-REM sleep, but it happens. And then we jump in here to dreams. So two theories as to why we dream are the consolidation theory, which basically says that dreams result from memories being consolidated. Your brain is processing information from the day into memories, and thus they appear in your dreams. Also have the activation synthesis theory, which is that dreams result due to the brain processing neural activity. It essentially makes a story out of it, and then your brain makes sense of the activity by making a story out of it to make sense. A little confusing. Short curve there. I'm not sure. No, or wait, no, yeah. Um, yes, it is possible to be in REM sleep without dreaming, according to uh some online AI. Yeah, that's right. Basically, you aren't get so REM is rapid eye movement, so your brain's active, and that's the place where dreams are most likely to occur, and chances are they will occur. Does that mean there's a guarantee they will occur? So I'd assume you could have REM sleep and no dreams. Chances are you are dreaming, you just don't remember it because a lot of people don't remember their dreams. And even if you do, you still probably don't remember what it actually was. So there are three theories as to why we sleep. Memory consolidation theory, same as why we dream, but a little different here. And so sleep allows for organizing and integrating memories. It's essentially a time for your brain to process and store all the information store all the information you got that day. The restoration theory, which says that sleep allows the body to restore depleted resources. And then conservation theory, which is basically just time to save energy. That's why you live so long, because sleep is needed in order for your body to conserve energy. We jump in here to sleep disorders. So there are five you need to know. So insomnia, difficulty falling or staying asleep. I think we all know what insomnia is. Sleep apnea, if so, a CPAP, you know what that is. That's what this is for. Sleep apnea is periods of when you basically so when you're sleeping, you stop breathing, you wake up, you resume breathing, and then you fall back to sleep. If you ever slept in the same room with somebody with it, it's pretty scary because they'll just be sleeping, they stop breathing, but then their body still knows, hey, wake up. You start breathing again, and then you fall back asleep. It's and the primary way that it's treated is with like CPAP, which is a constant positive airway pressure, so you don't stop breathing, which can actually sleep apnea can actually kill you if you don't happen to wake back up. That's pretty scary. Now, so you short cry with that. You still probably had dreams. Probably just didn't remember having them. Going along here, then you have narcolepsy. So narcolepsy is excessive sleepiness or falling asleep at random times during the day. It's really random, so you'll just be sitting there talking to someone and then you'll just fall asleep. But I'm really tired, and then you just fall asleep. And then you have somnambulism, which is just sleepwalking. It's when you get up and you walk while you're asleep. You can go have a snack, go to the bathroom, drive a car, but you're still sleeping. You're sleepwalking. And then you have rapid eye movement sleep behavior disorder, REM sleep behavior disorder, which is characterized by high levels of activity during REM sleep. So you're asleep, but you don't know any of this is happening, but because you're still asleep. But it's essentially a disorder where during REM sleep, muscle paralysis does not occur. It should occur. If it doesn't, kind of an issue. Basically, you're just you be flailing around in bed and you're still asleep, but because you don't have muscle paralysis during REM sleep, you're very active. That's it for sleep. Then we get to jump in here to sensation, which is pretty long here. 20 more slides, guys. So, sensation is the detection of stimulus energy from the environment. It is converted into neurochemical messages for the brain. So, detection is the point at which our sense notices the stimulus in the environment. Absolute threshold is the minimum amount of stimulus for us to notice it. The just noticeable difference is the minimum change in a stimulus that is noticed. Then we have Weber's Law, which states that two stimuli must differ by a constant percentage to be perceived as different. Roll something here. Be something here. Oh well. Basically, there's an example that basically says that so it differs by constant percentage. So, for example, if a shirt, I don't know, let's say a shirt is $90 and it's 10% off, but then this pencil over here that's $3, it's like 50%. It's like 50% off. Then basically it's basically a good example is as then you will naturally notice the 50% off as regard to the 10% off, even though that 50% off really doesn't matter. You still perceive it as different and as a deal per se. Basically, your senses do a lot of funky things, and there's nothing you can do to control it. And then you also come here to sensory adaptation, which is a decline in sensitivity to a stimuli after constant exposure. So, for example, if you live by a highway, your senses will naturally adapt to the noise, so thus you are less aware of the noise, or you become less aware of the stimuli. And then transduction, which converts stimulus energy into neural impulses. It allows the brain to interpret the information because your brain can only understand neural impulses. It can't understand stimulus energy. It can understand your senses. Yeah. Now we roll on in here to the sense of touch. So you have the ability to sense temperature, pain, and pressure. That's it. So temperature, which is two separate senses, you only feel warm and cold. There is no hot per se. There is no cool per se. You only feel warm and cold, and then those two interact, and then you can feel different temperatures as a result. And then pain, which is then followed by the gate control theory, which it's a theory that states the spinal cord has gates that either allow for or block pain. Gate control depends on whether or not you feel pain. Now then we have senses of the body. So the kinesthetic sense, which is the sense of one's body movement and position. It's info sent to your brain about the position of joints, muscles, and limbs. For example, a football wide receiver, you've seen them make those tiptoe catches. They have a heightened kinesthetic sense where they don't even look and see where their body is. They just naturally sense where their legs are and they know where they need to be. That's how they do it. And then the vestibular sense, which controls balance, and it's detected in the semicircular canals in the ear. That's not important. So, for example, Simone Biles would have a high or has a high vestibular sense because she's has really good balance. That's what controls balance. We come in here to gustation, which is our sense of taste, and so the five flavors we can taste are sweet, sour, salty, bitter, and umami, which is savory. Not sure it came up with umami, but it's kind of stupid. So, and then these five flavors are detected in taste buds on the tongue, and different individuals are categorized as different tasters based on how many taste buds they have on their tongue. You do need to know this. Super tasters have 35 to 60 taste buds per 6 millimeter. Medium tasters have 15 to 35 per 6 millimeter, and non-tasters have 15 or fewer per 6 minute per 6 millimeter. Try to hurry up and get this over with. We roll here to olfaction, which is our sense of smell. The lock and key theory. Basically to produce a scent, a particular fit is required between an odorant and a receptor cell. So if that odorant is the key, it unlocks the lock, which is the receptor cell, which then releases the smell of whatever it was. The pheromones, which are chemical messengers received through smell, and they're passed from individual to individual, and they affect others' behavior. It's partially attributed to why, so if somebody yawns near you, why you then also yawn. And then anosmia, something like that, is the loss or impairment of smell. Can't smell stuff? Oh well. And then sensory interaction here. So essentially, one sense combines with another to produce perception. So the sense of smell and taste work together to perceive flavor. Without one or the other, you don't really taste. When one of them is blocked, for example, you can't smell anything. Something you taste isn't going to taste the same, or isn't going to taste as good. Same thing, reverse way around. In order to taste, you have to use your sense of smell. This is how it is. And we roll right on in here to vision, which is our sense of sight. You are able to see, so the visible colors, you're able to see red, orange, yellow, green, blue, indigo, and violet. Any other color is some variety or mix of one of these seven. So the section of your eye you do need to know are so the lens, which essentially bends light and focuses it onto the retina. Your retina is the photosensitive surface of the back of the eye. It's what enables you to see. And the accommodation is when your lens changes its shape to focus objects onto your retina, near, far. It helps you to see near sight and far sight. And then we have the retina. So the retina contains rods and cones. The rods detect shape and movement, also black, white, and gray, while the cones detect color and fine detail. There are two theories of color vision. You have the trichromatic theory, which essentially states that the retina contains three color receptors, which are red, green, and blue, and those three combine to form different shades of color. Your eye mixes colors to then process that color. And then you have the opponent process theory, which states that the retina contains three opposing color receptors, those being red, green, blue, yellow, black, white, and that these opposing receptors enable vision of the other. Anything else there is mixed, of course. And then there are uh, color vision deficiencies are caused by damage or irregularities to one or more cones. Dichromatism is color blindness, partial color blindness. Two colors are perceptible. Most common there is red green color blindness. You only see red and green. Monochromatism, which is complete color blindness, and all you see is gray. Different colors or different shades of gray. And that's what this bottom thing is here. You have the original colors, and then you have the shades of gray there, which is kind of sad. And then you have your blind spot, which is the point where the optic nerve leaves the eye. Blind spot is where you can't see, essentially, so then your brain will then fill in the gap. and then vision deficiencies, which are often caused by damage to the occipital lobe, prosopagnosia, which is facial blindness, and you have difficulty recognizing faces. Basically, you can see the faces, but you can't put a name to that face. You don't recognize faces. And then you have blind sight, which is a blind person's ability to react to visual stimuli that they don't see. Like they can't physically see it consciously, but they believe they are seeing visual stimuli. It's something odd there. We have like three more slides. Let's stick it out here. Should be done here 10:30. So we have here audition, which is the sense of hearing, wavelength, which is the pitch or the tone, amplitude, which is loudness, and then auditory localization, which is the ability to identify where sounds are coming from. You want to figure out where the sound is. There are three theories of sounds. So the place theory, which states that the pitch depends on the location of the stimuli, which helps to explain high pitched sounds. The frequency theory, which states that pitch depends on the rate of nerve impulses. It explains low pitched sounds. And then you have volley theory, which is that pitch depends on which pitch is the rapid firing of nerve cells. It's a volley. And it creates the highest pitched sound. It creates the highest pitched sounds. And then this last slide here. So hearing difficulties slash deficiencies. So you have sensory neural deafness, which is damage to the inner ear or the auditory nerve. It is the most common type of hearing deficiency. And then you have conduction deafness, which is when sound does not reach the inner ear. It is caused by issues with the outer or inner ear, fluid, infection, earwax, a blockage that prevents those sound waves from reaching the cochlea. Well, you made it through. I don't care about all those questions there. This was biology. I believe next Monday at the same time we have cognition up. Um, yeah. I posted in the chat earlier. I posted again here in a second. But the lecture feedback form, please fill it out. This presentation. Give me a second. Please do fill that out, and then any questions, put them in the chat. I'll answer whatever you need. And for reference, I'm doing Unit 2 Cognition same time next Monday. You six left here. Any questions at all? And the presentation is linked on the schedule and it's further up in the chat here. It's been pretty good. Uh, yeah, school. Yeah, of course. Yeah, yeah. It's funny because I'm currently still in AP. I'm currently taking an AP Psych. This is basically a review for me too. I'm gonna go ahead and end the stage if there are no questions.